So good day. I've chosen 3 questions out of 5 in which the instruction states that I only have to answer 3. So the first question, which is the most interesting one, is how does the transposon allow the gene duplication or the DNA duplication in an individual? So let's create a fine line between cell division and transposon DNA or DNA sequence duplication. They are different because in the duplication or the cell duplication, the whole genome is copied. This is a much more controlled manner. While on the other hand, on the transposon's process of DNA sequence duplication, it just only duplicates segments and not the whole genome. There's a big difference between the two. So transposons are happening on a fully grown individual and not on the germ cells. Now, there are 2 types of transposons. The one that copies and pastes it, and the other one that just cuts it and pastes it. Now, there's a difference between copy and paste and cut and paste. Cut and paste meaning you're just moving. You're not copying it. It just moves. Now, the copying and pasting is that the DNA sequence stays on that loci, copies itself, and pastes it on another loci on the genome. Now, how does it work? Well, this transposon is specifically called as retrotransposon. This is the one that copies and pastes it. Retrotransposon. So how does it work? The transposon is transcribed by the intermediate RNA. So afterwards, what happens? It is reverse transcribed by the enzyme reverse transcriptase, which is synthesized by the transposon. Now, the reverse transcription causes the formation of a gene or a DNA sequence that is identical to the one that is staying on the original loci. Now, the copied one, which I'm referring to, is moved on another genome, and that is what we call duplication, since the copied one it remains on that specific loci and its copy is moved onto another loci or locus or a site or an area in a genome. Now, the other one is not deleted and the other one is pasted. So what do we call that? That is what we call duplication, since the process does not simply cut and paste it. Okay, so the process is simply copy and paste. So have you ever seen the documents that you have? You're copying it and pasting it into another, and you look back on the 2 files, you see 2 copies of that content, while cutting it and pasting it would cause the program to delete the other one and make the content exist on one side only. That is what we call cut and paste. Now, where does cut and paste happen? So cut and paste happen during the process of DNA transposon. This is the counterpart or the other transposon or another version of the transposon that does not copy and does not allow DNA duplication. So, how does DNA transposon happen? Why does it not copy the gene or the DNA sequence? Well, that's relatively simple. Since the transposase cuts the terminals and then drifts it into another loci. Now the direct copies or the direct sequences binds together, which makes the original location empty of that gene that moves. And the gene that moves, or the DNA sequence that moves, is pasted into another one, making this process just a jump and not a copy and paste. It is not a duplication. You just move the gene from one loci to another, or from one locus to another. Now, I apologize for the longevity of my explanation. I just want to express, or should I say, explain it in a more detailed manner based on how I understand it or understood it. So the next question is, how does the law of segregation affect the phenotype ratio? I'm sorry, because I'm just recalling the questions. So how does it affect the phenotype ratio? Well, it's relatively simple, because the law of segregation, there's a reason for why it is called a law. It is a law for a reason, since many of the scientists in the genetics community have agreed, or should I say all of them have agreed, which is why that theory became a law. Now, how does it affect it? Well, it happens during meiosis, particularly during prophase 1, anaphase 1, and anaphase 2. Now, why did I include prophase 1? Why? Now, suppose that these are two chromatids from different chromosomes. Let's say that these two are homologous chromosomes. Okay. Now, the portion of this one is transferred onto this one. Okay. Now, the portions are segregated, which is why I counted prophase 1 as a part of the segregation process. Okay. Portions are segregated. So that is the beginning of segregation. Now, those segregated portions are then separated on the process of anaphase 1. So the homologous chromosomes separates. And when they separate, there's a big chance that most of the traits are distributed. Moving on to the anaphase 2, the sister chromatids now separates, and making your traits more reduced, because you're relying on a recombined sequence, and plus you are obtaining the trait from your father or your mother. Another 50% of it would come from your father or your mother. Now, given that the segregation process is already hectic, or already, let's say, complicated, now, combining the two, or let's say, your father has a segregation process, your mother has a segregation process, which makes the overall outcome based on chance. But how does this chance break? Well, this chance is highly disrupted by the dominant trait or the dominant allele. So let's say that the sister chromatid that you have on the anaphase 2, let's say that the gametes that you have on the anaphase 2 of your mother contains the blue eyes recessive trait. Now that's a lot of process coming down to that recessive trait. Let's say that your mother's germ cell or gametes have given you the recessive trait of blue eyes. Now another 50% would come from your father. What if your father as a result of that segregation process, he may be a carrier of the blue eyes, but due to the segregation process, the trait that have been transcribed to that gene is the dominant trait, which makes you a big letter B and a small p. So that is how the law of segregation affects the phenotype. Phenotype ratio, or the phenotypic ratio. I'm sorry for the terms. Phenotypic ratio. So it is all about chances. Chances of what would be the sister chromatid that would be fused during fertilization. Now moving on, let's say that you have a dominant trait, but the phenotype or the phenotypic ratio does not fully determine what phenotype you have, because keep in mind that the phenotype of a person is, or the trait of a person is not only dependent on the gene, it is also dependent on the environmental factors, or should I say affected by environmental factors. So for example, when we go into detail, a person that has the gene for tallness, for a tall height, you have the gene or the allele for the tall height that is expressed on you. So if you have the allele for a tall height, but your environment is scarce of nutrient, meaning you don't have that much access to nutrients, well, it doesn't matter if your allele is tall when you lack the nutrient to support it or to express it even more with the cooperation with the environment. You wouldn't be tall, since the nutrient wouldn't be adequate enough to support that allele. So that's why the genes is not always followed, since the environment also affects it. Moving on to the last part, or the last question that I would like to answer. I'm really sorry for prolonging this video. I'd like the third question is, can survival of the fittest be related to the rules of gene inheritance? Yes, survival of the fittest can be related to the rules of gene inheritance. Why? So we inherit the genes of our parents, so we are the offspring. Now our sons and daughters would be our offspring. Now we are adapted on this environment, in this specific geographic area. We are adapted to this. Therefore, our offspring would be adapted, genetically adapted in this environment, which is why the survival of the fittest in a particular geographic location is highly relevant on the inheritance of genes. So, take for example, I, as a person, as an individual who lives in the Philippines, suddenly goes to Africa and manage to move there and stay there. What do you think would happen to me? My body is not fully adapted to that location. Therefore, it would cause me to survive not better or lesser than the others. Okay, so African people would survive better than me, since they are adapted on that location. So the survival of the fittest also is relevant in a way that inheritance, following the rules of segregation, which creates variation, would make another specific portion of the population survive better than the other. Why does another why does some of the population survive better than the other? Why do they survive better than the other? Well, because it is due to the variation. Variation that is bounded to the rules of inheritance. Why? Say for example, I've increased the temperature in the ecosystem. Let's say that the world's temperature is increased by about 10 degrees Celsius. 10 degrees Celsius may not be significant enough, but looking at it globally, those people that are highly adapted on warm environments would survive better than us in the Philippines that is not highly adapted on high temperatures. Since what temperatures do we have? We have 40 to 60 range. So yeah, so the population living on the or adapted on the locations that are warm or hot would survive better if that happens, if the temperature suddenly rises globally. So variation is related to survival of the fittest, and survival of the fittest is related to adaptability or the survivability of a person globally and geographically. That's all. Thank you, and I do apologize for the time that I've consumed.